Wheat is one of the most important staple crops worldwide. On average, it provides about 20% of the protein, starch, fiber, vitamins, minerals, and essential amino acids. Apart from determining the quality of bread and pasta, wheat proteins can induce inflammatory reactions in a small fraction of humans, such as celiac disease, wheat allergy, and non-celiac wheat sensitivity. Thus, the proteome profiles of different wheat species are of high relevance for stakeholders along the supply chain to produce healthy products. During my PhD research employing large-scale proteomics, thousands of proteins across hundreds of samples were quantified. First of all, an optimized data analysis pipeline was designed to deal with the big proteomics data. This was necessary to estimate a multitude of parameters such as heritability, which is used to determine the relative influence of environment versus genetics on the expression of a protein. Secondly, to gauge the variation of a protein across tested cultivars. And also, this was necessary to implement different kinds of statistical procedures. Additionally, the lab methodology was improved by introducing a novel hybrid approach that measures the proteome by label-free quantification while providing absolute quantification of the key amylase trypsin inhibitors. Comparing the proteomes of spelt and common wheat, about 3,000 proteins were quantified. About 400 of the giant proteins between spelt and common wheat were statistically significantly different. Importantly, the negligible difference in the abundance of allergenic proteins suggests that spelt and common wheat might not be different in causing wheat-related intolerances in humans, confirming other studies. In a subsequent study, we examined the proteomes of 6 flours and 14 bread types from spelt, common wheat, and rye. Results showed that the allergenic proteins were more prevalent in spelt or common wheat than rye, and grain type was likely more important for allergenicity than milling or fermentation technique. Moreover, we generated the flour reference proteomes of 5 wheat species, namely spelt, common wheat, durum, emmer, and einkorn. In any pair of wheat species, more than 50% of the proteins differed significantly. Particularly, the total amount of all allergens and only amylase trypsin inhibitors was 5 and 7 times lower in einkorn than in common wheat, respectively. This highlights the potential of einkorn as an alternative cereal crop for people with sensitivities to wheat allergens warranting further clinical trials. In the final study, it was found that the environmental conditions have a big influence on the expression of proteins. Majority of the proteins showed low heritability and only a small percentage were consistently expressed in different field conditions. However, hundreds of proteins with high heritability were also found, including important ones for human health and baking quality. It should now be possible to specifically manipulate the expression of these proteins by selecting and breeding for superior cultivars along the supply chain, thus enabling the production of safe and healthy products.